Dear students, today let us discuss on level of structural organization in proteins. The spatial arrangement of atoms in protein is called its conformation. The possible conformation of a protein includes any structural state without breaking covalent bonds. A change in conformation could occur by rotation about single bond in polypeptide chain. Hence, theoretically, numerous conformations are possible in a protein containing hundreds of single bonds. The conformation of protein exists in thermodynamically the more stable, which is having the lowest gift-free energy. Proteins in any of their functional folded conformation are called native proteins. Now, primary structure of protein. Primary structure of protein or first level of protein structure deals with the sequence of amino acid residue and spatial arrangement of atoms in a polypeptide chain, which is linked by a covalent peptide bond. The backbone of polypeptide can be pictured as a series of rigid planes sharing a common point of rotation at alpha carbon. We can read as CC-NC, CC-NC, and CC-NC, and so on. The first carbon and the last carbon is alpha carbon. Here, the alpha carbon of each adjacent amino acid residue are separated by 3 covalent bonds. In the figure, the first covalent bond is formed between first alpha carbon and carbon, and second covalent bond is in between carbon and amide nitrogen, the peptide bond, and the third covalent bond is in between amide nitrogen and second alpha carbon. In this series of plane, X-ray studies found that there are 6 atoms directly or indirectly participate to the peptide bond. Here, the 4 atoms, namely alpha carbon, amide nitrogen, and the second alpha carbon, are in the main chain, and 2 atoms, which is oxygen atom of carbonyl group and hydrogen atom of amide nitrogen, in the peptide. Altogether, the 6 atoms lie in the same plane, means they are coplanar. In the peptide bond, second bond is in the figure. The oxygen atom of carbonyl group and hydrogen atom of amide nitrogen are in the trans configuration. That means oxygen atom is in the up position, hydrogen atom is in the down position, and the oxygen atom of carbonyl group has partial negative charge and nitrogen atom of amide has partial positive charge. Hence, it set up a small electric dipole. Partial sharing of these 2 electrons occur between carbonyl oxygen and amide nitrogen. Therefore, the peptide bond is in double bond character with a bond length of 1.32 angstrom, which is shorter than alpha carbon to amide nitrogen bond with a bond length of 1.45 angstrom. Since the peptide bonds C-N bond has double bond character, the rotation of such peptide bond is strictly prohibited. However, the first covalent bond between alpha carbon and carbonyl carbon and third covalent bond between alpha carbon and amide nitrogen is purely single covalent. Hence, these bonds are free to rotate. By convention, the angle of rotation between alpha carbon and amide nitrogen is called phi, and angle of rotation between alpha carbon to carbon is called psi. The phi and psi angles of each amino acid allow protein to fall in many different directions. Now, the secondary structure of proteins. The secondary structure of protein or second level of protein structure deals with the common regular folding pattern of a polypeptide or protein. These structures are mainly arise from hydrogen bonding between electronegativity oxygen atom of one peptide bond and amide nitrogen of another peptide bond, either in the same or different polypeptide chain. 4 types of secondary structure are found in proteins, Let us discuss them one by one. Number 1. Alpha helical structure. Alpha helical structure is a rod-like structure. In this structure, the polypeptide containing rigid peptide bond is tightly wound around an imaginary axis drawn longitudinally in the middle of the helix, and the R groove, either the side chain of the amino acid, protrude outward from the helical backbone. Generally, the length of alpha helix is 10 to 15 amino acid residues. The repeating unit in a single turn includes 3.6 amino acid residue, and the distance between corresponding point per turn is 5.4 angstrom along the axis. Alpha helix structure is stabilized by hydrogen bonds in the main polypeptide chain. Particularly, the CO group of each amino acid N-terminant forms a hydrogen bond with the NH group of the amino acid that is situated 4 residue ahead, means towards the C-terminant in the sequence. All the CO and NH group form a hydrogen bond except those amino acids which are close to both ends of the helix. Alpha helix may be right-handed if the helical structure appears screw type or clockwise, or left-handed structure, it is counterclockwise conformation. Essentially, all alpha helix found in proteins are right-handed. The right-handed are energetically more favorable because the steric class between side chain and the polypeptide backbone is less. The alpha helix content of each protein is widely differ and may range from 0 to 100%. For example, ferritin, an iron-binding protein, contains 75% of amino acid residue as alpha helix. Single alpha helix is usually less than 5 angstrom. However, 2 or more alpha helix can interlink to form a very stable structure which can have a length of 1000 angstrom or more. For example, myosin and tropomyosin in muscle protein and keratin in hair are made up from interlink of alpha helix. Now, factor affecting the stability of alpha helix structure. Stability of alpha helix structure is mainly affected by amino acid sequence in the alpha helix region. For example, the presence of long negative charge glutamic acid or positive charge lysine in the polypeptide chain will repel each other, hence affected the helix conformation. And presence of proline and glycine causes constraint in the alpha helix, where proline residue in the peptide linkage introduce a kink in a alpha helix, hence cannot form any hydrogen bond with others. Number 2, beta pleated sheet structure or second type of secondary structure of protein. In this structure, the polypeptide chain is extended in a zigzag, therefore they are relatively flat. This zigzag chain can be arranged side by side to form a structure resembling series of pleats, and R groups of adjacent amino acid protrude from zigzag structure. 2 or more beta pleated sheets are layered close together within a protein. The layered polypeptide chain may be parallel or antiparallel. In parallel, the adjacent polypeptide segments are in the same orientation, whereas in antiparallel, the adjacent polypeptide segments are in opposite orientation. In parallel arrangement, the CO group of each amino acid is form a hydrogen bond with NH group of the 2 amino acid residue further along the adjacent polypeptide segment. In antiparallel arrangement, NH group of each amino acid is form a hydrogen bond with the CO group of adjacent polypeptide segment. In beta sheets, many structures, typically 4 or 5, but as many as 10 or more, may organize together. Such beta sheets can purely antiparallel, purely parallel, or mix. Generally, beta pleated sheets contain high amount of glycine and alanine residue, which has the smallest R group. For example, fibrin protein of silk and spider web are rich in glycine and alanine residue. Number 3 is the beta turn, or also known as the reverse turn. Beta turn structure is the connecting element which link the ends of the two adjacent segments of antiparallel beta sheets. The structure is 180-degree turn that involves 4 amino acid residue and also known as the reverse turn. In this structure, the oxygen atom of CO group of first residue form a hydrogen bond with the nitrogen atom of NH group of the fourth amino acid. However, the peptide group of central 2 residue do not participate any inter-residue hydrogen bonding. Glycine and proline residue often occur in the beta turn, where proline makes the tight turn. Beta turn structures are mostly found in near the surface of the protein, and the central 2 residue which do not participate any inter-residue can form a hydrogen bond with water. Number 4 is the loops. Loops are random coil-like irregular structure found in protein. Loops are well known for reversal of polypeptide chain and comprises of 2 to 16 amino acid residue. They are generally found to connect alpha helix and beta sheet structure. Similar to beta turn, they are found near the surface of a protein, thus participate in interaction between protein and other molecules. Now, the tertiary structure of protein, the third level of protein structure. Each protein is made up of variety of helices, beta sheets and non-regular region, which are folded in a unique 3-dimensional structure. The overall 3-dimensional structure of protein is referred to as tertiary structure of proteins. This folding of protein into native 3-dimensional conformation includes not only the position of primary backbone atoms, but also includes all the interaction between side chain atoms of distant places. The interaction is just an application of solubility rule like dissolved like. The stability of tertiary structure of protein is maintained by many non-covalent interactions in their side chain in addition to covalent peptide and disulfide bond. Non-covalent interaction include hydrogen bond. Hydrogen bond may be formed between 2 alcohol groups, for example, serine and threonine, or between an alcohol and acid, for example, serine and aspartic acid, etc. Number 2, hydrophobic bond. The hydrophobic bond may be formed between nonpolar side chains, for example, leucine and isoleucine. Number 3, van der Waals forces. The van der Waals forces may be formed between tightly packed hydrocarbon side chains. And number 4 is the electrostatic force or salt bridge. It may be formed between positive and negative charge of side chain, for example, lysine and aspartic acid. Now, let us discuss how R groups are interacted in the tertiary structure of water-soluble globular proteins. For example, myoglobin. Myoglobin is an oxygen-binding muscle protein. Myoglobin is a water-soluble complex globular protein made up from 153 amino acid residue. X-ray diffraction studies show that about 70% of main chain is folded into 8 alpha helices and the rest of the chain forms a turn and loops between helix. It is very compact structure and almost no empty space inside. The interior of the myoglobin is made up of nonpolar residue such as leucine, valine, methionine and phenylalanine. However, charged residue such as aspartate, glutamate, lysine and arginine are absent. Only 2 histidine residue found to contain in the interior that play a critical role in the binding of iron and oxygen. Polypeptide chain therefore folds so that its hydrophobic side chains are buried inside and its polar charged side chains are on the surface. The secret of burying a segment of main chain in the hydrophobic environment arises from pairing all the Ni and CO group by hydrogen bonding in an alpha helix or beta sheet, and unpaired hydrogen bonding in peptide Ni or CO group markedly preferred water environment. The hydrocarbon side chains are tightly packed and stabilized by the van der Waals forces, which requires intimate contact. But in the case of the hydrophobic protein, for example in porine, an outer membrane protein of many bacteria, the distribution of hydrophobic and hydrophilic amino acid residue are reversed in compared to water-soluble protein. Now another structure, domains. Domains are structurally independent folding units looking like a separate globular proteins, but they are all part of the same polypeptide chain connected in the primary structure. Large proteins often have 2 or more domains, and the size of the domain range from about 30 to 400 amino acid residue. For example, troponin C, a protein found in muscle, has 2 domains which are present in one polypeptide chain, and extracellular part of the CD4, the cell surface protein, which the ICIV attached, has a 4 domain. Now quaternary structure of protein, it is a 4th level of protein structure. Many proteins contain 2 or more separate polypeptide chains, and each polypeptide chain is called a subunit. The arrangement and the nature of their interaction of such 2 or more subunits in 3-dimensional conformation is known as quaternary structure of protein. The protein with only one chain have no quaternary structure. Therefore, quaternary structure of protein deals with only the proteins which have more than one polypeptide chain. Quaternary structure is stabilized by non-covalent interaction such as hydrophobic interactions, electrostatic interaction, hydrogen bonds, as well as covalent disulfide bonds. Now naming of the quaternary protein. Quaternary proteins are named as dimer for 2 subunits, trimer for 3 subunits, tetramer for 4 subunits, and so on. And if the subunits are of same kind, then prefix homo is used, and if it is different kind, then called hetero. Example, hemoglobin is made up of 4 subunits with 2 different kinds, hence known as heterotetramer, where 2 subunits of one type alpha and 2 subunits of another type beta. Now in conclusion, the primary structure of protein or first level of protein structure is the sequence of amino acid residue in a polypeptide chain. Secondary structure of proteins or second level of protein structure is a common regular folding pattern of a polypeptide chain due to the hydrogen bonding between electronegative oxygen and nitrogen atom of adjacent peptide bonds, either in the same or different polypeptide chain. Secondary structure includes alpha helical, beta sheet, turn, and loops. Tertiary structure of protein refers to the overall 3-dimensional conformation of a whole polypeptide chain in its folded state. Tertiary structure is stabilized by many non-covalent bonds, viz. Hydrogen bond, hydrophobic bond, salt bridge, and covalent disulfide bond, etc. And the arrangement of 2 or more subunits in 3-dimensional conformation is the quaternary structure of protein.